In this activity, you'll examine three fundamental plant cell types that make up simple tissues: parenchyma, collenchyma, and sclerenchyma. These cells form the basic building blocks of plant organs such as stems, leaves, and fruits, and provide functions including storage, photosynthesis, flexible support, and rigid support. Plant organs are made of multiple tissues, and many of those tissues are built from three fundamental cell types. Parenchyma makes up the bulk of most plant organs and consists of cells that are generally isodiametric, meaning they are similar in length and width. These cells may be spherical, oval, round, polygonal, or elongated in shape. They have thin cellulose cell walls and may be tightly packed together or separated by small intercellular spaces. Parenchyma cells perform a variety of essential functions, including photosynthesis, storage of food and water, and secretion. Collenchyma occurs in layers just beneath the epidermis of dicot plants, either as a continuous band or in patches. The cells have unevenly thickened walls, especially at the corners, due to deposits of cellulose, hemicellulose, and pectin. They may contain chloroplasts, lack intercellular spaces, and provide flexible mechanical support to growing parts of the plant such as young stems and leaf petioles. Sclerenchyma is composed of dead cells with thick lignified walls and pits, providing strong mechanical support. It occurs in two main forms: fibers, which are long, narrow, thick-walled cells often found in bundles, and sclereids, which are shorter, irregularly shaped cells with very small lumens, commonly found in hard or gritty tissues such as seed coats and the flesh of fruits like pear. These three fundamental cell types may occur as simple tissues when only one cell type is present. Later in the lab, you'll see how these same cells can also be components of complex tissues such as xylem and phloem, where multiple cell types work together for transport. Before you begin observing the microscope slides, take a moment to think about this question. Why would a plant need parenchyma, collenchyma, and sclerenchyma instead of relying on a single type of support cell? Think about flexibility, strength, growth, and the different environments plant organs experience. Pause the video and record your prediction in your lab notebook. The first sample that we will look at is celery petiole, where we will view parenchyma and collenchyma. The celery petiole is the stalk that connects the leaf blade to the stem. Its job is to hold the leaf upright and position it for light capture, while also allowing the leaf to bend in the wind rather than snap. Because of this, the petiole needs two different types of tissues working together. Parenchyma cells make up much of the internal tissue. These thin-walled living cells are involved in storage, metabolism, and maintaining turgor pressure. They also allow for some flexibility. Collenchyma cells are found just beneath the epidermis, especially in the ridges of the celery stalk. Their unevenly thickened cell walls provide extra strength, but because the walls are not lignified, the tissue can still stretch and bend. Together, parenchyma and collenchyma allow the celery petiole to be both supportive and flexible, strong enough to hold up the leaf, but not so rigid that it breaks under mechanical stress. As you observe the celery slide, think about how the location and structure of each tissue type reflects this balance between support and flexibility. Begin by viewing the prepared celery petiole slide under medium power. At this magnification, focus on the overall organization of the tissue. Notice the outer epidermis and the ridge regions just beneath it. Look for areas where cells appear elongated and more tightly packed near the edges, and areas where cells appear larger and more loosely arranged towards the center. Collenchyma cells are found just beneath the epidermis, especially in the ridges of the celery stalk. They appear as clusters of lighter gray, elongated cells with unevenly thickened cell walls, particularly at the corners. It's hard to distinguish where the cell wall of one cell begins and ends. I've also labeled the interior of the cell as the cytoplasm. These thickened corners provide strength while still allowing the tissue to bend without breaking. Parenchyma cells are found more centrally in the petiole, but they're also found between the collenchyma and vascular bundle. They are larger, more irregular in shape, and have thin primary cell walls, which are easier to see. I labeled the cytoplasm inside the cell. Intercellular spaces are often visible between them, reflecting their roles in storage, metabolism, and sometimes photosynthesis. Pause the video and draw at least 3 parenchyma cells and 3 collenchyma cells. Label the cell wall, cytoplasm, and identify each as either parenchyma or collenchyma. Focus on accurate proportions and wall thickness rather than artistic detail. The next sample that we will look at is pear tissue, where we will view sclereids. Pears are mostly soft, juicy fruit tissue made of parenchyma cells. However, when you eat a pear, you may notice a slightly gritty or sandy texture. That gritty texture comes from clusters of sclereids, also called stone cells, embedded within the soft parenchyma tissue. Sclereids are a type of sclerenchyma cell with very thick lignified cell walls. Unlike parenchyma and collenchyma, these cells are dead at maturity and extremely rigid. In the pear, sclereids provide mechanical protection for the developing seeds and help reinforce the fruit tissue so it does not collapse under its own weight. Their hardness also discourages herbivores from chewing directly into the seed-bearing region. As you observe the pear slide, think about how the thick walls and small lumens of sclereids reflect their protective, supportive function, very different from the thin-walled flexible cells you saw in the celery petiole. Sclerenchyma tissue includes 2 main types of cells, fibers and sclereids. Fibers are long, narrow cells that occur in bundles and provide strong tensile support. They are commonly found in stems, leaf veins, and around vascular tissues, where they help the plant resist bending and breaking. Sclereids, also called stone cells, are much shorter and more irregular in shape. They have extremely thick lignified walls and are often found in hard or gritty tissues such as seed coats and the flesh of pear fruits. There are different shapes to sclereids. Both fibers and sclereids are dead at maturity and function primarily in support and protection, but they differ in shape, location, and the way they reinforce plant tissues. In this lab, you will directly observe brachysclereids in pear tissue as a representative sample of sclerenchyma. Now we are switching to the prepared pear tissue slide. Under low power, look for coarse, gray, grainy clusters. These clusters are sclereids, also called stone cells. You may notice the larger, thin-walled cells as well. These are cells that we covered earlier. What do you think they are? If you said parenchyma cells, you would be right. At higher magnification, each sclereid appears irregularly shaped with very thick lignified walls. The central cavity is the lumen. Thin lines extending through the wall are pit canals, which allow limited communication between cells. Pause the video and sketch 1 or more sclereids. Clearly label the thick cell wall, the lumen, and the pit canals. Using your observations and background knowledge, answer the following. Describe 2 ways in which sclerenchyma differs from parenchyma. Consider the cell wall thickness, cell function, and whether the cells are alive or dead at maturity. Identify the characteristics of collenchyma that set it apart from either parenchyma or sclerenchyma. Think about the cell wall structure and the type of support provided. Fibers and sclereids are both types of sclerenchyma cells. How are they similar to each other? How are they different? Think about the shape, location, and function. Pause and record your responses. This activity demonstrates how different simple tissues are specialized for different roles. Parenchyma provides metabolic and storage functions. Collenchyma provides flexible support in growing regions. Sclerenchyma provides rigid protective support. Once again, the central theme holds. Structure supports function, from thin primary walls to heavy lignified secondary walls.